Hello everyone, this is Andy from Med School EU, and today we're going to finish up our first lecture with the last topic, the role of enzymes. So here's just a brief description of what we're going to learn about enzymes and how they interact in organisms. So first we're going to look at an overview of what enzymes are and what do they do. We're going to look at enzyme kinetics, enzyme inhibition, and allosteric enzymes. So let's first begin with an overview of enzymes and what do they do and what are enzymes. So typically enzymes are biological catalysts, meaning that they speed up reactions, chemical reactions in the body, but they're not consumed by the reaction, they're not used up by the reaction, or if they are used up, they are regenerated by the end of it. So as you can see here, they're not consumed, and almost all enzymes are proteins. Some could be natural RNA molecules, some have catalytic properties. However, most enzymes in general, they are proteins. Okay, and enzymes are critical for biochemical reactions in all living organisms. So now let's take a look at why we need enzymes in the first place and what do they do in our body. So typically reactions occur spontaneously in our bodies, and a lot of those reactions do not need anything for them to happen. So they happen on their own without enzymes, without catalysts. However, they occur at a very slow rate, and therefore our bodies and other organisms cannot sustain themselves if they are producing products at a very slow rate, 0.1 of a product per second, let's just say. However, there are two primary ways to increase the speed of reaction in order to meet our demands. So if the demand is, let's say, 1,000 of a product per second, this is not even close to what it would produce spontaneously, right? So what we need is a way to speed up the process of gaining this much product per second. It will happen eventually, but not in a second spontaneously. So there are two primary ways to do that. One is by increasing the temperature of the reaction. So let's analyze why we cannot simply just increase the temperature in our body. So our core temperatures are, in humans, 37 degrees Celsius, right? And typically if our core temperature increases even a couple of degrees, that means we're sick. So, and typically at 42 degrees Celsius or more, the human body starts shutting down, and or organisms start failing, and the person dies. So why does this happen? Well, because proteins, enzymes, and other structures, they can only be functional at a certain temperature, and that's the 37 degrees Celsius. That's when they are in their shape. But however, when it even increases by just 5 degrees, those enzymes and those proteins that are in our body, they denature, right? They denature, and therefore our organs can't function, and we have a full system failure and the person dies. So another way to increase the speed of reactions is going to be through enzymatic catalysis. And these are enzymes that do not increase the temperature, but they facilitate the formation of products, and they do it at a much faster pace. So the products would form 10 to the 5-fold or even 10 to the 7-fold on average. So that's how much they speed up the rate of the reaction. Where they're not consumed, they do not increase the number of products, they simply increase the speed at which the products are made. And it's important to note that enzymes are also very specific for their substrates. So they cannot simply bind all kinds of reactants together. They are only specific for certain substrates. And there are specific enzymes for each type of a reaction. All right, so now let's take a look at a diagram and really see how the reaction occurs and what type of effect the enzymes have visually on a diagram. So first of all, let's mark down some of these terms here. That's delta G, and that's simply the free energy of activation. So here we have the reactants, here we have the products, and this is the reaction as it goes. So in order for the reactants to become the products, they must undergo a certain state, and this state is called the transition state. In the transition state, the reactants come between something between a reactant and something between a product. They're right in the middle. So from this point on, they can go either this back to the reactants, or they can proceed and the reaction occurs and they become the products. But this is the critical part where the reactants must cross in order to become the products. And this is called activation energy. That's the energy needed for the reactants to be activated in order for them to proceed with the reaction and become the product. So here we call this delta G transition state, and same with this one, delta G transition state. Now this one, the big diagram here would be without no enzyme, so no catalysis, and this line right here is going to be with enzyme. All right, so let's see what happens. Without the enzyme, look at how much more energy is needed for that reactant to become the product. Now, with the enzyme, what happens is it lowers the activation energy. It facilitates for the activation energy to go down, and it's much easier to convert the reactants into products, and therefore it occurs at a much faster pace. Now note that the amount of products that's formed does not change. The same thing occurs in both types of reactions. Nothing changes. The amount of product is formed the same. However, the reaction, the speed of it, happens a lot quicker because of this lowered activation energy. So here I have the definition of delta G, and it is the amount of energy needed to convert 1 mole of reactant from the ground state, so this is the ground state, to the transition state right here. So that's the delta G that we have marked here, and as you can see, it's significantly lower. It's significantly lower here with the enzyme present. So again, in summary, just what we just learned about the diagram is that enzymes increase the rate of the reaction. It happens faster because it decreases the activation energy, so it's easier to form the products from the reactants. And enzymes do not change the position of the equilibrium. So if we are to make 1 mole of product A, it does not suddenly increase the amount of product that's made. It's simply made faster, at a faster rate. So let's take a look at an example that I drew of what kind of happens with enzymes. So this is an enzyme and what they sort of look like. I mean, they're all in one way or another are different. However, this is just a general outline of how it occurs. So here we have an enzyme in white. And as you can see, this enzyme has this active site, so we can label this active site. Active. And this active site binds the substrate, so this would be the substrate. And they can perform and facilitate all kinds of reactions. So this substrate has one unit of, let's just say this is a disaccharide, and there's one monosaccharide, another monosaccharide. This is 1, 2, right? So we got 2 disaccharides, and they should be broken down into just a monosaccharide so the body can use it for energy, like 2 glucose molecules, for example. And what happens is they make contact with this active site, and this facilitates it to be able to be broken down a lot quicker, and they go their separate ways as monosaccharides. All right, so next let's talk about enzyme inhibition. And what are inhibitors and what do they do to enzymes? Well, inhibitors are exactly what they are in the name. They inhibit the enzyme activity. So they're compounds. They could be proteins, they could be other enzymes, but they're compounds that bind to the enzyme and they decrease enzymatic activity, right? So what occurs is that the speed of the reaction decreases because of these inhibitors. And for example, this is typically how drugs and toxins actually work. If you've seen, movies with snake poisons and things like that, how those poisons work is they enter the body and they inhibit certain enzymes, and then the body cannot produce and facilitate a certain reaction fast enough, so then organs begin to shut down and it causes system failures. And this process could be reversible or irreversible. It depends on the drug, it depends on the toxin, it depends on the molecule that inhibits these enzymes. So here are the 4 common types of reversible inhibition that we will discuss, and not to great detail, but just to give an overview in case it comes up on the exam. So the common types are competitive inhibition, pure non-competitive inhibition, mixed non-competitive inhibition, and uncompetitive inhibition. And we'll take a look at each one more closely. And first, let's begin with competitive inhibitors and what those are. So structurally, competitive inhibitors look and resemble normal substrates. They look exactly the same, they have the same kinds of shape. They could differ a little bit in terms of their structural abilities and their functions. However, they are able to bind to inhibitors in exactly the same way, in the exact same active site as the substrates that we need for the reaction. Now, the binding of the inhibitor and the substrate to the enzyme is exclusive, and that's the main point of competitive inhibitors. They exclusively bind to the enzyme, meaning that only the inhibitor or the substrate can bind to the enzyme. So if the inhibitor binds to the enzyme, then the reaction will not proceed. But if the substrate binds to the enzyme, then reaction will proceed. So it is going to be a game of the concentration. So the concentration of inhibitor versus the concentration of the substrate. Whichever molecule exists at a higher concentration will win and will be able to proceed and form products. Or if there's more inhibitors, then more enzymes will be disabled and the reaction will not proceed. So let's take a look at what these things look like on an enzyme. So here we have an illustration of an enzyme that's in yellow, and the substrate right here, it binds to the active site and the reaction proceeds. Reaction proceeds. Right? However, the same similar type of molecule that would be the inhibitor could also bind to the enzyme, and if there's more inhibitor than the enzyme, this will typically happen more often with the enzyme, and then there is no reaction because the inhibitor now occupies the active site, and the substrate, even if it just comes in, if the substrate just comes in here and tries to bind, it won't work. It won't happen because that is taken up by the inhibitor. All right, next let's discuss the two types of non-competitive inhibition. So there's the pure and there's the mixed, and we're going to decipher which one is which and how non-competitive inhibition works in general. Well, basically, the difference between competitive and non-competitive is that in non-competitive, they're not competing for the active site. So both the inhibitor and the substrate can bind simultaneously to the enzyme, which means that they both have two different binding sites. The substrate binds at the active site and the non-competitive inhibitor binds somewhere else. Now in pure non-competitive inhibition, the inhibitor binds far away from the active site. So it could bind further away from the active site, and what it does is that it changes the enzyme conformation, which then makes the enzyme less effective in catalysis. So it can convert less substrate in less amount of time, or it can just permanently stop the enzyme from working because it changed the enzyme conformation. And remember, enzymes are typically proteins, and proteins work with structure. So if structure is not there, the protein will not work. Right? So if it changes the enzyme conformation, it changes its structure, and typically the substrate would not be able to be converted to the product. Now in mixed non-competitive inhibition, what we have is something very similar, and it's just an inhibitor that binds close to the active site. It binds right beside it, typically right by the active site. And what it does is it affects the affinity of the enzyme for the substrate. So it may not change the conformation, it may not stop the enzyme from working entirely. However, it changes the affinity. So the binding of the substrate is less because this affinity is changed by this mixed non-competitive inhibitor that binds close to the active site. And the final type of enzyme inhibition that we're going to discuss is called uncompetitive inhibition. And this type of inhibition occurs when the inhibitors bind to the enzyme substrate complex. And what this means is that the enzyme has already binded with the substrate, forming what is known as enzyme substrate complex. And once this complex is formed, only then the inhibitor binds. And what occurs is no reaction, because this inhibitor has bonded with the ES complex, the enzyme substrate complex. So the reaction simply does not proceed. Now what it looks like on the diagram is that we have an enzyme, we have the substrate. So first, this is number one, the substrate binds to the enzyme, and this binding occurs successfully in the active site. However, this uncompetitive inhibitor comes in and also binds to the enzyme and the substrate, which makes no reaction. It prevents the reaction from happening because now this enzyme is typically blocked, and it just kind of stays in the enzyme without being turned into products. All right, and the final topic that we're going to discuss in terms of enzymes is going to be allosteric enzymes and what they are, what they do, and how they work. So allosteric enzymes, they consist of multiple subunits, and they're binding of effector molecules that results in a conformational change. Conformational change. That's the final thing that occurs with allosteric enzymes. And they typically do not follow the similar curves like Michaelis-Menten kinetics if you have done biochemistry before. However, what happens is they affect the enzyme kinetics meaning that they affect the rate at which enzymes work, either positively, being the activators, or negatively, being the inhibitors. So there's two types of effector molecules. You could have activators, or you could have inhibitors. And each one can have an effect on the enzyme and its kinetics in terms of how fast it produces its products. So allosteric enzymes, they have an active site, but they also have one or more regulatory sites. And these regulatory sites is exactly where the effector molecules bind. Either it's activators or inhibitors, they bind to these regulatory sites, whereas the substrate continues to bind to the active site as usual. So let's take a look at this diagram here. And what we have is that the middle here, there's no effector. That means the allosteric enzyme is going to proceed its reaction in a normal rate as it typically would without any molecules affecting it. But when the activator comes along, it positively affects enzyme kinetics, which means it produces more substrate at a faster rate. Now, if we have the negative effect, which is the inhibitor binding to the regulatory site of the allosteric enzyme, what we get is a slower rate of substrate production and the ultimate lowering of the number of substrates being produced as the final product. So now let's take a look at an illustration of what of how this occurs in enzymes. So here we have this molecule right here would be the effector. This one right here is the substrate. Now, obviously, we know that this right here is the active site where the substrate binds, but this little spot right there is going to be the regulatory site. And the regulatory site is where the effector binds, whether it's positive activator or negative inhibitor. The effector molecule will bind to the regulatory site. It does not interfere with the active site. Now, there's two things that could happen depending on whether it is an activator or an inhibitor. So here we have the first one that occurs, and that's it binds. So the effector molecule binds to the regulatory site, and what it does is it increases the affinity of the substrate. It increases the affinity of the enzyme for the substrate, meaning that the substrate would bind to the enzyme more readily. And that's because this is going to be an activator. Activation. Because this molecule is an activator. It is an effector that activates the enzyme. And the other scenario is going to be if this effector molecule affects it negatively, and it would be an inhibitor. So this is an inhibition. And what happens in this scenario is that once the effector molecule binds to the regulatory site, it changes the conformation of the enzyme, and it cannot bind the substrate. The substrate does not bind it. It is out of there. It cannot bind to the active site because of the changed conformation due to the binding of the inhibitor, which is the effector molecule that binds to the allosteric enzyme. So this takes us to the end of chemistry of living things. We have completed that one. And the next lecture, we will talk about the cell as a basis of life, and we will begin with the topic of cell theory and cell size.